Thanks for tuning in to a journal club today. My name is Jennifer Deyo. I'm a pharmacist and medical science liaison with Natera's Organ Health Division. This is a really timely and much needed article, thankfully published by the American Journal of Transplant, on the opinions of stakeholders or African American patients about the use of apolipoprotein L1, or what I will be referring to as APOL1, genetic testing in the realm of living kidney donation or renal transplantation. If you're newer to the subject of APOL1, it's important to know that this gene is actually very helpful to have in the human genome. It's part of the innate immune system, which is always helpful, as that is going to help us fend off pathogens that we would come into in our natural environment. We know very specifically about APOL1 that it helps monocytes differentiate into macrophages. We start to see this come into play in the transplant and nephrology space because we now know there's coding alleles G1 and G2 that are actually associated with chronic kidney disease, a heightened risk of actually going on to develop end-stage renal disease in people with African ancestry. 13% of African Americans residing in the U.S. at this point are postulated to have pathogenic, or what we would call problematic, APOL genotypes. And again, doing a full picture on why APOL1 is so important, it really lies in the fact that this actually helped you when you were living or are living in Africa to prevent African sleeping sickness. So this is a parasitic infection that is transmitted from the tsetse fly. And what happens is, after basically an initial viral type illness, this pathogen actually goes on to invade the central nervous system. And at that point, you're going to get inversion of the sleep-wake cycle, hence the name African sleeping sickness, and undergo a lot of other very detrimental mental status changes, as well as gait and speech disturbances, and if left untreated, will lead to coma, seizures, and death. Other very intriguing parts of APOL1 are that we know in vitro we see HIV-1 partially inhibited, and that is through the effects that APOL1 has here on this GAG protein. So it kind of helps the virus get targeted for degradation. Now, this isn't quite as clear when we look at the gene as far as in vivo results. So this is a meta-analysis of over 2,000 African American patients from 4 pretty large prospective trials in looking at opportunistic infections in HIV and AIDS when you had APOL1 variant alleles. So again, in vitro, we know there's some straightforward activity going on, but as we see here on the right side of the screen, there's really no difference in any viral, any parasitic, mycobacterial, opportunistic infections. We did see a trend toward reduced infections with fungal opportunistic infections. So more to come here. The study design here was representative of the African American community. Sample study, it used a Qualtrics reward program to include adults with African American ancestry, had to be fluent in English and reside within the United States. The recruitment took about 4 days for this study, and they did survey questions for clarification in 25 people. They used the Think Aloud technique with Amazon Mechanical Turk, or MTurk, which is a way to help you distribute and perform tasks virtually. It was, again, a national study. The IRB was approved from Beth Israel Deaconess Medical Center in Boston, Massachusetts. There were 780 eligible survey responses, but 63 individuals did have to be excluded because of not answering truthfully. They had a nice attention filter question that wasn't answered appropriately in 49 patients, and basically what this means was that you had to actually type out the word yes, Y-E-S, and enter. If you didn't do that, it was deemed that the attention wasn't what it needed to be to answer a lot of pretty particular questions in genetics. And then if you completed this survey in less than 5 minutes, you also got excluded. Despite these exclusions, it's really important to know that we had a really good group of respondents, 585 people to be exact. 76% of them were female, and a variety of ages, but most commonly patients were on the younger side, 18 to 50 years old. Thankfully, all of the OPT regions were included. Only 7% of the respondents reported that they had a past medical history of renal disease, although 32% of them did report someone in their family did have renal disease. 82% of the patients identified as being African American. 83% reported they had a low risk of health literacy problems as defined by needing help when interpreting literature, brochures from their physician or their pharmacy. As far as less than or equal to 50% of renal disease being due to genetics, 74% of the respondents said that this was the case. Most respondents, 78%, said renal disease after kidney donation was or should be the same or less than non-donors. 69% of patients, importantly, were not aware of APOL1 testing. As far as results, basically what we looked at here as far as a potential living kidney donor, taking a free APOL1 test if it was available, they basically put together, I would probably take the test or definitely take the test, and combined these two numbers here to say 61% of patients would undergo APOL1 testing as a potential kidney donor. Now, something that's really important here is that 89% of patients would like a deceased donor kidney to be tested for APOL1 before accepting the organ. As far as more results here, from predictors of high likelihood of wanting APOL1 testing as a potential living kidney donor, what we found was that older age, over 35 years, was statistically significant. If you were in the Southeast or the Midwest, you were also more likely to want to be tested at 0.03 on that p-value. And then last but not least, if you highly identified yourself as African American, you were more likely to want to have yourself tested. And then if you perceive that you could have a higher risk of kidney disease after being a donor, you also would more likely want yourself to be APOL1 tested as a donor. So here, if you were interested in genetics or had a high self-efficacy in genetics, it was correlated with wanting this genetic testing to be done. And then if we kind of flip the role here, and we say older age and higher interest in genetic counseling correlated with the interest in having your donor organ tested for APOL1. So this would be if you were actually receiving the organ, not donating it. So again, if you were greater than or equal to 35 years of age, and if you had a high interest in genetics. So again, this article couldn't come soon enough, and many thanks to the authors and AGT for publishing it, because, and I quote here, these perspectives differ from what is currently done in practice. So right now in the transplant community, we are not testing kidneys for APOL1, and we're not all the time is it standard practice for us to test a donor that's of African ancestry. So this is a needed starting point to including the stakeholders being African American patients in APOL1 testing. So the question for us today is, how would including transplant recipients and donors add to or change this study? And then, could we see genetic testing impact which organs are accepted or declined? This is obviously a pretty tough topic because we know that organs are just such a very precious resource. But at the same time, definitely need to listen to people of color. And since this is going to affect not only them, but family members and donors. So it's never too early to mention that in the interim, as we are going forward about the Apollo study, okay? So this is APOL1 long-term kidney study. This is a national study, and it's actually going to tell us a lot about outcomes, but the results are not going to be in until 2023. So I would just urge you to think about when you were talking with donors and recipients that are of African ancestry, to think about this study that included almost 600 participants. Most of them, again, only 7% that didn't even have renal disease, and the interest in wanting to know more about the genetics and predispositions for kidney disease. Thank you so much for your time.